Cells and control. Chromosomes. The nucleus has your genetic information in the form of chromosomes, which have coils of DNA. Genes are short sections of DNA that code for a protein's control characteristics. There's 23 pairs of chromosomes in each body cell because you inherit 23 chromosomes from your mother and 23 from your father. So you have 46 chromosomes in total in all cells, but sex cells. Sex cells, also known as gametes, only contain 23 chromosomes. A diploid cell has 46 chromosomes, whereas a haploid cell has half the number of chromosomes compared to a diploid cell, so only have 23 chromosomes. A good way to remember the difference between haploid and diploid cells is that haploid starts with a H, which could represent half, and diploid cells starts with D, which could represent double number of chromosomes compared to haploid cells. Cell cycle. Cell cycle is what happens to the cell, so it can divide, and these new cells can be used for growth and repairing damaged cells. 1. Interphase, and this is when the cell grows, organelles replicate, and increase in size and DNA is replicated. 2. Mitosis is when chromosomes line up at the equator of cell and cell fibers pull each arm of the chromosomes from each side of cell. 3. Cytokinesis is when two identical daughter cells form, when cytoplasm and cell membrane divide. Mitosis is an important part of asexual reproduction because this type of reproduction only involves one organism to produce offspring by replicating its own cells. Mitosis produces two daughter cells, each with identical sets of chromosomes to parent cells, because sets of chromosomes in daughter cell's nucleus are the same as in parent cell's nucleus. Mitosis produces two genetically identical diploid daughter cells. Steps of mitosis. 1. Prophase. This is when the nuclear envelope breaks down and the chromosome are free to move in the cell. 2. Metaphase. This is when the chromosomes line up in the middle of the cell. 3. Anaphase. This is when spindle fibers are formed and attach to the center of chromosomes, pulling half the chromosome to one side and the other half to the other side. 4. Telophase. This is when a nuclear envelope forms around the two different sets of chromosomes. Growth, differentiation, and cancer. Growth in animals is because of cell division and differentiation. Cell division happens by mitosis, and after these cells differentiate to specialized forms specially adapted to their function. Example is cells of muscular system, which allow movement, and cells in circulatory system are specialized to transport substances. Most cells in animals differentiate early on and then don't differentiate again. Most specialized cells make more of the same cell by doing mitosis. But red blood cells don't have a nucleus, so can't divide and are replaced with adult stem cells, which are one type of cell that doesn't differentiate early on. Another example of a cell that doesn't differentiate early on is embryonic stem cells. Growth happens by cell division and differentiation in plants, and they can also grow through a process called elongation. Plants grow in a certain direction by absorbing water into vacuoles, and this is controlled by substances like auxin. In plants, a lot of cells differentiate throughout the plant's life. They only differentiate when they reach their final position in plants, but they can still redifferentiate when it's moved to another position. Cancer happens because of small changes in cells called mutations that lead to uncontrolled cell division, which produces a group of cells called a tumor. Monitoring growth using percentiles. Important to measure growth of organism, and this could be used for crop growth or babies when they're growing. Use percentile charts to tell us the rate of growth of an organism. Growth of babies measure using mass, length, or head circumference. Baby born at 50th percentile for mass is heavier than 50% of babies. Baby born at 25th percentile for mass is heavier than 75% of babies. Baby born at 75th percentile for mass is heavier than 25% of babies. Stem cells in embryos, animals and plants, and benefits/risks of their use in medicine. Stem cells are undifferentiated cells which divide to make more cells, so some differentiate to have different functions. Type of stem cells. 1. Embryonic stem cells form when egg and sperm cells fuse to form zygote. Differentiate into any type of cell and body. Scientists clone cells and direct them to differentiate into almost any type of cell in the body. These are used to replace insulin-producing cells in people who have diabetes, new neural cells for diseases like Alzheimer's, or nerve cells for those paralyzed with spinal cords. 2. Adult stem cells, found in bone marrow and form many types of cells including blood cells. 3. Meristem in plants, found in root and shoot tips, differentiate into type of plants and have this ability throughout life of plants. They're used to make clones of plant. This is used if the parent plant has certain desirable features like disease resistance for research or save rare plant from extinction. Therapeutic cloning involve embryo made with same genes as patient. Embryo produced are harvested to obtain embryonic stem cells, grown into any cell patient needs like new tissues or organs. Advantages are that they don't reject because they have exact same genetic makeup as the person. Benefits of research with stem cells, used to replace damaged or diseased body parts. Unwanted embryo from fertility clinics used because if not, they will be discarded. Research into process of differentiation. Risks of research with stem cells. Don't understand process of differentiation, so it's hard to control stem cells to form cells we desire. Removal of stem cells leads to destruction of embryo. People might have religious or ethical objections. If growing stem cells are contaminated with virus, an infection can transfer to the person. Money and time could be better spent on other areas of medicine. Structure and function of nervous system and reflex arc. Nervous system lets us react to our surroundings and coordinate actions in response to stimuli. 1. Receptor cells convert stimulus into electrical impulse. 2. Electrical impulse traveled down the nerve cell called sensory neuron to central nervous system, which is the spinal cord or the brain. 3. Then the impulse is sent along the relay neuron, which connects both sensory and motor neurons together. 4. Information is processed and appropriate response is coordinated, so electrical impulse sent along motor neuron to effector. 5. Effectors do response, which can be muscle contraction or glands secreting hormones. Neurons have an axon, dendrites, and dendrins. Autonomic response is involuntary like reflexes and are important because they stop a person from getting badly injured because the information travels down reflex arc, so important responses happen quickly. This is different to normal response to stimuli because impulse doesn't pass through conscious area of the brain. 1. Stimulus detected by receptors. 2. Impulses are sent along sensory neuron. 3. CNS impulse passes to relay neuron. 4. Impulses sent along motor neuron. 5. Impulse reaches effector, so response is done. An example of a reflex is when the pupils dilate to avoid damage from bright light, and another example is moving your hand from a hot surface to prevent further damage. Synapse. Synapses are gaps between neurons. 1. When an impulse reaches the end of a neuron, a chemical called neurotransmitter is released into the synapse. 2. The neurotransmitter diffuses across the synapse. 3. Neurotransmitter reaches the next neuron and triggers an electrical impulse to form again. Different neurotransmitters have different effect on frequency and speed of the impulse in the second neuron. Some nerves are myelinated, meaning they're surrounded by a myelin sheath, and this allows nerve transmission, also known as an action potential, to travel faster as the nerve cells are insulated. Myelin sheaths are made of Schwann cells. Thank you for watching. Please like, comment, and subscribe.